Esteemed speakers, ladies and gentlemen, good afternoon and a very warm welcome to this second in the monthly Brain Matters series organised and hosted by the Human Brain Project. My name is Katrina Sickle, I'm an independent moderator, and I have the privilege for the next 60 minutes of helping to bring to life today's discussions on networks for consciousness and medical use cases. Now, our aim with this series is to present in a very dynamic and a very accessible way the results, the brain research, the brain results, and the methods from within the Human Brain Project. And our audience, you good people, really is a very diverse one. We range on the one hand from the molecular biologist, and at the other end of the scale, we have the curious non-specialist, such as myself. Now, in today's edition, you are going to hear from two renowned neuroscientists who are both working in the field of consciousness. They are Marcello Massimini, who is a consciousness specialist based at the University of Milan, whose research focuses on understanding the changes that take place in the thalamocortical networks when consciousness fades and recovers, such as when we sleep, when we wake up, or during anesthesia. Our second speaker today is Pieter Roelsema. He is director of the Netherlands Institute for Neuroscience, where he and his colleagues examine how the human brain makes awareness, perception, movement, social interaction, and other cognitive functions possible. So his research focuses on optogenetics, which involves using light to switch brain cells on and off. Now, the great thing about our webinars is that you get to pose your question to our expert guest speakers. And the way that you do this is simply go into the private chat on the social media platform from which you are joining us today and type in your question or comment. Please, super short, say to whom the question or comment is addressed, and we will start to pose that at around just after halfway through our webinar. So get your thinking caps on, please. And of course, at any time, you can tweet words of wisdom to the wider community, #BrainMatters. So let's get stuck in. I've got somebody to join me to frame this week's discussions, and I'm very delighted to welcome her now. Her name is Mavi Sanchez-Vives. She is ICREA Research Professor at the Institute of Biomedical Research in Barcelona, where she leads the System Neuroscience Group. So I'd like to ask her to please join me on screen now. Hello, it's a great privilege to have you with me. Thank you. You have a very illustrious background there. Now, let me get stuck in, please, immediately. You are the leader of Work Package 2, entitled Brain Networks, Underlying Cognition and Consciousness. And I'd like to chat a little bit with you about the subject for this second edition in our Brain Matters webinar series, and of course, specifically the work that you're doing in the Human Brain Project. So the obvious first question is in a nutshell, what is the objective of this work package, please?
Yes, so the objective of this work package is to implement a data-driven, complex computational model that carries out cognitive tasks while expressing different brain states. So let me explain to you what this means. So let's think of a cognitive task, for example, object recognition, which seems to be quite easy for us, but we want a model based on data to perform this task. So this is actually quite complex because it requires stimulus processing, change detection, learning, memory, decision taking to see which is the right object. So this requires the integration of activity of different cortical areas and so on. So we want to have a network that can perform these cognitive tasks, but at the same time expresses natural brain states. And what do I mean by this? Well, it can sleep, or it can be awake, or have the activity corresponding to these patterns, because in this way it's going to express natural physiological dynamics, synchronization, and is going to be able, for example, to consolidate information during a sleep and so on. So something very important in a project of these characteristics where it's very collaborative. So in this group, we are 38 research groups, and it's very important to be able to do more than we can do independently. And what we can do more is that we can study many different levels. And what this means is that we can integrate people working on the same problem, but they can be working either on apical or on dendrites, or on single neurons, or small circuits, or very large circuits integrating different areas, or the whole brain activity. So what is very challenging and very attractive is that by this collaboration, we can actually cover all these very different levels studying the same problem. So what we would aim to reach ideally is that we have this model that can perform some cognitive tasks, but can also express these different patterns of activity that can correspond to wakefulness or attention, but also pathological situations, like, for example, disorders of consciousness, like a coma, or anesthesia, and or brain injury.
And that's interesting what you say there in terms of why it's particularly special, the Human Brain Project. You've got a lot of people working together, coming at this from different sides, and that work can help nourish each other. You called it challenging in a good way. I just want to focus on this word consciousness, because is that not something, for want of a better word, a little bit slippery for science? Is it a bit more philosophical?
Yeah, this is very interesting, and actually you are right. I think until the 1990s in mainstream neuroscience, it was almost taboo to study consciousness. However, since then, there has been an exponential increase in the number of publications. And what we are focusing here about consciousness, and we are going to hear about this today in the next talks, they are aspects of consciousness that they are not spiritual or esoteric or philosophical, even they are measurable. So what are those two aspects of consciousness? I will mention very briefly. So one of them is the consciousness that we lose when we fall asleep, or when we have a few drops of anesthetic in our blood, we suddenly vanish. Our consciousness vanish. We stop being, in a way. So consciousness levels, therefore, they can vary because we fall asleep, or because of drugs or anesthetics, but also because of brain lesion. There can be disorders of consciousness. So we will hear about this. And what we are interested in is to be able to capture, to measure the level of consciousness by the brain activity. We will hear about this to Marcello Massimini, and the reason why this is important is not only because it helps us to understand what is important in the brain in order to maintain and to generate consciousness, but also is very critical for patients that are unresponsive to be able to measure whether they are conscious or not. And then, very briefly, I will tell you about the second aspect that we are going to hear to Peter Roxema speaking about conscious perception. So it's a different aspect of consciousness. When do we become conscious of a stimulus? So we know from visual illusions, for example, that sometimes we can have a stimulus in front of us and we don't perceive it, and eventually it emerges. So what does it change in the brain? And what is very important also is that we are going to hear about what are the clinical applications of these basic research questions.
And that's very important. And that's why, of course, in the title, we have medical use cases. And we will, of course, find out more about that when both of these gentlemen have spoken about their work. I talked there, I was sort of saying, I asked a question on whether it was rather philosophical. You've talked about measurable, so it's very clear where you're coming from in the project. But what about the ethical aspects? The ethical aspects of consciousness. From what you tell me, these studies involve many ethical issues. I mean, you've got determination of consciousness. You're now identifying decisions of minimally conscious patients. So how are you dealing with this within the project?
Yes, so the ethical considerations and neuroethics have been quite very relevant in the Human Brain Project. So in particular, in our work packets, we have a team expert on ethics and philosophy, And I think it's very important for them and for the scientists to work side by side to be able to determine what are the conceptual, the social, the ethical dimensions of our research. And it's important to determine this from the moment that this research is being done. So we need to develop the right philosophical and ethical frameworks for this research of cognition and consciousness. And in these two topics that we have mentioned about consciousness, there is one which is how to ascribe. What are the criteria to ascribe?
I found this on the web for researchers.
Very sorry. The criteria to ascribe consciousness, for example, as we have said, to patients, but eventually also to machines, for example. So what are these criteria to ascribe consciousness, which are theoretical, they are empirical, philosophical. And there is another aspect which I think is very relevant. We are going to hear about measuring brain activity and about stimulating the brain. And it's very important to consider what are the ethical implications and how this is going to be regulated and so on in the future.
Well, I think it's very encouraging from what you say is that you've got these groups working side by side, the ethics, the specialists, you've got the researchers. It's not something that comes in at the end of the process. And I think that's very important to underline here, in particular about this project. A last question, please, before we will hear from the first of our guest speakers. I would imagine that there is so much work going on this topic in general around Europe. So the obvious question to ask you is what makes it different? You've touched upon that, but what makes your work in the Human Brain Project different in any respect?
Yeah, so as I mentioned, of course, there is a lot of very good research going on, and most of us have also collaborations outside. But of course, the difference, as mentioned, is that there is this collaboration that allows this multi-level approach, which is impossible to do when working independently. Also, it's very important to have this close collaboration between experimentalists and modelers, but something very important, I think, that we are trying to do in the Human Brain Project is work also side by side with engineers, with ICT, and we are working in the development of a platform called eBrains. And in this platform, when we do our research, we are currently sharing the data, we are sharing the analysis pipelines, we are sharing the models, and this is going to be available for the community. So this can be used in the future or currently actually as well. So it can be used by many groups of people, like not only neuroscientists, they can be used by experimentalists to be able to use pipelines to analyze their data or to do simulations, or by students to learn to deal with real brain data, or by data analyzers, or by clinicians to run models and simulations. So this is...
I need to ask you to round up now. Okay. Sure. No, thank you. Just let me catch, because I interrupted you there. You think...
Yes. So we believe that it's going to be very relevant for the community, not only for the neuroscience community, but also for the medical community and so on.
Okay. Thank you so very much. Sorry to interrupt you, but you are coming back. So I'm going to give you the opportunity to take a back seat just for a moment while we hear from our two neuroscientists who are going to present the practical work and then the practical applications. And I will invite you back to join us later. So thank you very much for jumping into the hot seat right at the outset. See you soon. So where are we? We're right at the point where I can ask our first speaker to join us. We've had the background. Let's really hear about that work that has just been profiled. So who is our first guest speaker? It is Marcello Massimini of the University of Milan. And he is going to tell us about his work, which focuses on the development of tools to better measure consciousness in non-responsive or minimally responsive patients. So if I can ask him to please join me, that would be lovely. Hello.
Hey, Katrina. Hi.
Hello. So you've got a splendid background as well. Yours is real. Mavi's was actually a brilliant green screen background.
Yeah, this is real.
You are actually really at work. So we may have issues with your sound. Let's keep our fingers crossed we don't, but I'd like to ask you to just dive in, please, and talk about your work and how the project helps you in your science. Thank you.
Thank you, Katrina. Do you see a brain?
I do see a brain. I see your project. I see your slides. Yes.
It's a good start. All right. So, of course, we have understood a lot about this very peculiar physical object. We understand the neural chain of event leading for, say, visual motor coordination, memory encoding, color discrimination. We know these processes so well that we can implement these functions in artificial circuits also, but there is still a mystery. And the mystery is, as Mavi said already, is that sometimes the electrical activity of the brain changes and we don't exist anymore. The entire universe collapses as far as we're concerned. And this is very deep as a mystery and also very familiar because this happens every night. So now understanding the mechanism of loss and recovery of consciousness is not only a scientific challenge or an existential conundrum, but also a practical matter. And again, as Mavi said, we focus on this practical aspects in HPP. And for example, when we deal with patients, we want to sense consciousness in brain injury patients. Since we don't know the internal mechanism of consciousness, we treat the brain as a black box, as you see here, and we work on input and output. Squeeze my hand, open your eyes, and we observe behavior. But sometimes patients are disconnected on the output side because of lesion to motor systems. Other times they're disconnected on the input side because of lesion to sensory systems. And in this case, this method can fail. So as a matter of fact, we are now dealing with islands of consciousness because we are saving a lot of brains with intensive care, which are difficult to assess, brain islands. And these islands of awareness are really challenging from the perspective of an external observer to judge. And this is really a clinical issue because patients' potential in these conditions are many, the size of a small city. So what does it feel to be a big chunk of tissue with billions of active neurons disconnected from the external world? It is difficult to say. What we know, though, based on our own experience, is that consciousness can be generated within the brain, even in condition of sensory motor disconnection. This happens every night when we dream. So for this reason, really, we have the ethical and clinical urgency of developing measures that are based on the internal state of the circuits above and beyond sensory processing and motor output. But unfortunately, this turns out to be quite challenging. As we all know, we have reviewed the problem recently. Empirically based markers are not so easy to find and not so reliable. So here, theoretical neuroscience can help. So one example is this paper that came out in '98, and this was the first attempt to relate consciousness to network circuit complexity in the brain. It was further developed, but the general basic idea is very simple. So you take experience as we experience it. The properties of experience are that it's very rich and unitary at the same time, integrated and differentiated. And then the idea is that, well, if I have to look for consciousness in the brain, I look for these properties in the circuit. That is a balance between integration, differentiation, unity, and diversity, in a word, complexity. So this is the idea. Metaphorically speaking, is like, we want to make sure that the orchestra of the brain is made of different elements that are able to interact tightly to form an integrated whole. And what you would do with an orchestra is to ping the orchestra and just listen to the music, the symphony, the complex symphony of consciousness, possibly. In the brain, we do sort of the same. So we kickstart a subset of neurons, and then we engage these internal interactions, and we measure the information content, the complexity of this music, electrical music produced by the brain. We can do this by stimulating non-invasive the brain with TMS and by recording the response with EEG. So now the prediction is that when consciousness is absent, you either have local responses because the system is not integrated, it's not unitary, global responses that are simple and stereotypical because the system lacks differentiation. And the prediction, the fundamental prediction, is that when you end consciousness, you must have this balance between unity and diversity in the system, and thus responses that are spatio-temporally complex. So this is what we want to measure, and these are the predictions. And then, over the years, we've been testing this prediction in many different conditions. So on the left side, red box, you see conditions where consciousness is present. We know because subjects are there, they can tell us wakefulness, brain injury patients who are able to report, or we know that because people wake up from REM sleep and ketamine and say, well, I was dreaming, hallucinating. So this is our benchmark conscious condition. You see high complexity in these traces. On the right side, you have loss of consciousness in sleep, non-REM sleep, and anesthesia. So this is a calibration in known conditions, sort of a ground truth where we calibrate the measure. And then the idea is to apply the measure in the large gray area of patients who are not able to report, unresponsive patients. So before doing this, of course, you need to develop, measures of complexity, and we are doing this within HBP. One way to do so is to take your brain response, then you analyze the spatiotemporal dynamics, and then you binarize this dynamic space and time to come up with a binary file, sort of this binary image. And the idea is that you can compute the complexity of these interactions by zipping it. So the more you can zip, the less complex it is. And then you can put this value called perturbational complexity index on a scale, and you can put your calibration. So this is our calibration, our ground truth.
You can even slow down a smidgen because you just zipped through the zip bit there, but just so we can follow your story, go ahead. I know you're conscious of time. Carry on. So you talked about zipping it for differentiation. Just go back to that.
I mean, zipping is, a way to measure complexity and, to explain, the rationale. If the response is local, no integration, it's easy to zip. If it's global, but there is no differentiation, it's very redundant. You don't need a lot of information to zip it. It's like when you zip images. If the response is rich and global, then you have a hard time compressing it. So this is the idea, right, of the zipping. And you come up with one value that is compressibility, if you want. It's called technically algorithmic complexity. And then you plot the value, and these are a reference scale based on the measurement we did in patients in subjects who are able to report. And then based on this distribution, you can draw an operational cutoff. And the idea is that if you have a complexity that is high, is in the range of the conscious benchmark, then even though the patient is unresponsive, there must be something there. So when you test it in patient, it works very well in minimally conscious patients. These are patients where consciousness is fluctuating, very difficult to detect, and we have a very high sensitivity. But then what is most important, we can draw inferences in the patient where you have nothing to say behaviorally, vegetative state patient, the one that are fully unresponsive. And here it's interesting to see that you can stratify them in low complexity patient, the majority of them, but then 20% of vegetative state patients, they have internal brain complexity in the circuit that is comparable to the one of the conscious benchmark. So here, the inference is that there must be some kind of disconnected consciousness, or at least you have to treat them like this. So this is it's interesting, but what's most important, I think, is that within and beyond HBP, we are all converging on this idea that complexity matters for consciousness. So these are works from Paris, from Liège, beautiful works, and also from groups outside of HBP, suggesting that really the best way for the moment to assess consciousness based on brain-based measures is this measures of integration, differentiation, information sharing, or complexity, all revolving around the same concept. And I think this is very encouraging, and we are in a good place here. In practice, what do we do when we have such measures? So take two patients that are non-responsive, so they're behaviorally identical, and with a measure based on brain circuits, you can open the black box, and maybe you find patients with high complexity, and here that there must be a potential for consciousness, and what you can do in these patients is to restore channels of communication, assuming that there is consciousness there, and you can do this with brain-machine interface, and Peter will talk more about that. And in these patients, instead, on the right side, where you have low complexity, well, this is a challenging, proposal, but what we need to do is to restore complexity whenever possible.
Okay.
This is important.
Okay. I need
Yep.
to ask you to slowly round up, but I will. Go ahead.
I'm done. This is important because, some patients are stuck in low complexity, other recovers, and we need to understand what are the keys to turn the neuronal mechanism. And this is, what we do in HBP now in this third phase of the project. So we have collected data about, why complexity is fade and recovers. about the neuronal mechanism at different scales, from slices to animals to humans. And now we have to combine this, taking advantage of the eBrain platform, into models in order to have predictions about what are the manipulations that we need to know to do to restore complexity. And we have already 2 ideas, about mechanisms that might play an important role. One is silencing the neurons by stability, the new tendency of neurons to become silenced. And another one is dendritic decoupling. This is discovered by Matthew Larkin within HPP, and it's a very interesting idea. So I think that working on this, we can make a difference for consciousness science, but also for clinical applications.
Thank you. And as I said, I am an interested non-specialist, and it sounds critically important. And I'm going to say, silly, without making light of this in any way, all of those Hollywood films, when you see people, waiting by bedsides and fingers to move or eyelashes to flicker, what's so fascinating and what you're explaining in these measurements is you're going way above that. I mean, it's so important. Tell me, what are the general implications? If you had to sort of sum it up, what are the general implications for your work within the project?
Yeah, so you give me the opportunity to pull up this figure that I like a lot. So this is a figure graph by Christoph Koch originally, which says that, for the moment, we assess consciousness based on what we can observe from an extrinsic perspective of function, intelligent behavior. This might work in nature, but we are changing nature. So we have these patients which are disconnected, and by the point of view of intelligent behavior, we cannot observe much. And then we are building machines that are very functional, very intelligent, and they're becoming more so, like AlphaGo and others. And this is becoming challenging because, we can miss consciousness where this is present, or see consciousness where it is not present, going back to what Mavi was saying. So we need to work more on this axis, finding on the y-axis, finding better measure of consciousness above and beyond function. So I think this is a major challenge that we have, and which has also a cultural value in general, an educational value, developing a sensitivity to consciousness.
And just as a close, which is important, we do talk medical use cases. So if we're talking about disseminating into a clinical setting, very short, what sort of time frame are we looking at for that?
Well, I think we are now changing, these techniques are quite complicated for the moment. TMSEG should be developed towards a more, easy technique like ultrasound echography, bedside measure. We are working towards that. I see 5 years to develop that, but also all together in HPP and beyond HPP, we should develop a pipeline, a workflow, putting together all the best measures that we have developed for better diagnosis, prognosis, and treatment. And we are working towards that, really.
Thank you. Now, Marcello, there's been a lot of questions coming in as you've been speaking. So what I'm going to do is I'm going to let you take a back seat, and then you can have a chance to look at the private chat, because you'll see some of the stuff that's coming in. Have a look at that, and I will introduce our next speaker, and then I'll have some questions for you afterwards. But thank you so very much, and for condensing all of that into such a short time. I know it's not easy. So thank you. We will hear more from you shortly. Okay, so while Marcello is relaxing, I'm going to invite our next guest speaker to come and present, and then I will invite both Marcello and Mavi back for a chat. So I'd like to now invite to join me Peter Roefsema, the director of the Netherlands Institute for Neuroscience. So if he could come on screen. Hello, sir. Very nice very nice to see you again. Now, when we had a chat before this webinar, and I was saying, what's a good way to describe the link between Marcello's work and yours? And you suggested that he's the kind of the bystander, because he's reading from the brain, and he's trying to develop tools to pick up consciousness where there is some doubt, whether there is any. You're not reading from the brain, you're writing to it, creating conscious perceptions. So can you tell us more about that work, how you calibrate computer-brain interfaces to ensure electrical stimuli inputs reach the level of attention? Thank you very much. The floor is yours, sir.
Yes, so
voila, I can see your screen. Great. Okay, go ahead.
Yeah, so today I would like to talk about how a stimulus may reach or may not reach consciousness, and the same stimulus sometimes makes it and sometimes doesn't. And I would like to talk also about the potential application of these findings. So in the world, there are about 40 million people who are blind, and many of those will not benefit from treatment in the eye because the connection between the eye and the brain is lost. So for those people, a logical approach would be to make an interface with the visual cortex. And people have started working on that, and that's also what we're working on in my team. And if you stimulate electrically a group of neurons in this region of space, actually it's what we call retinopy. So it's basically a layout of the entire visual field in the visual cortex. If you then stimulate one of those at one of those locations, one group of neurons, a subject, and it can also be a subject that has been blind for a number of years, will see a dot of light, and it will be a dot of light at that location in the map where you're stimulating. Now the goal is to have many electrodes there, thousands or even more, so that you can create thousands or more dots of light at different spatial locations. Okay, so and now you can work with this like a matrix board along the highway. If you switch on one bulb on the matrix board, subjects will see a dot of light, but you can also create a pattern by switching on some of these electrodes. Okay, and that's the eventual goal, a goal that scientists have long had. Now, this is the system that we would really like to build. So there will be a camera that takes in images from the outside world that then needs to be translated into brain stimulation patterns that should be meaningful, as you see here. They then need to be transferred to inside the brain. So there should be a brain chip that then talks to electrodes that are going to electrically stimulate the neurons. Okay, so that's the system that we want to build. Now, the thing is that if you stimulate with a very weak current, you are in a situation where the stimulus may or may not reach conscious awareness of the patient. Okay, so that's an important question. And a few years ago, we carried out a study where we looked at that issue. And here we first started with visual stimuli. So all the audience, I would like to invite you to look at this red dot in the middle of the screen. And what I'm going to do is to present to you a stimulus that sometimes can be very weak. So now I'm going to present the stimulus, 1, 2, 3. I suppose that most of you saw the stimulus. Now I'm going to do it again, 1, 2, 3. Okay, so now it was more difficult. Try it again, 1, 2, 3. And the last time, 1, 2, 3. Okay, now my suspicion is that many of you did not see all these stimuli. And what we found in the study is that if you present a weak stimulus over and over again, sometimes if it's close to the threshold for perception, sometimes it will, and sometimes it will not reach consciousness. So what happens here? So what we found is actually that the difference is well explained by a theory that was proposed by Stanislas de Hane a couple of years ago, who called this theory the global neural workspace theory. And what happens here is that if you present a weak stimulus, it needs to be propagated to higher areas of the brain in a sufficient strength. Now, if you do so, then there is a process that he called ignition. And ignition actually causes a reciprocal interactions between different brain areas, such that if you take the stimulus away, it's a very weak and brief stimulus, that the activity pattern is upheld through these interactions. We call it a working memory. Okay? Now, if you now present the same stimulus, in some conditions, it may not reach the higher areas in sufficient strength. Okay, if you then take the stimulus away, it will fade out and it will not be perceived. It will fail to reach consciousness. If the same stimulus, and that is due to the noisiness of the brain, kind of makes it out of the noise, then it can reach the higher areas and it can cause ignition, causing this working memory trace. Okay, so this is background knowledge that we can use in the design of a visual cortical prosthesis, because we're also stimulating these brain cells very weakly. And we're testing this now in monkeys, and we implanted in the monkeys more than 1,000 electrodes. This is what it looks like. So here are all the electrodes. Each of these squares actually corresponds to 64 electrodes. And we're now going to stimulate in primary visual cortex, the first cortical stage of visual information processing, in recording from the next higher areas to see whether what we stimulate here, whether it's strong enough to cause conscious perception. Okay. Now, what we do here in this graph, on the x-axis here, we're stimulating with different current strength. And here on the y-axis, you see the hit rate, the probability that the monkey perceived the stimulus. And you see that if we stimulate strongly enough here in this case with 20 microamperes, the monkey always sees the stimulus. If you then reduce the strength of the stimulus, there's a threshold here, 12.6 microamps, and then the animal reports it about 50% of the time that we give the stimulus. Now, the interesting thing is we also had electrodes in a higher area, area V4, through which we could record. And what we observed is that if you give weak stimuli, we basically see no activity in area V4. If we give stronger stimuli above a certain threshold, then we start to see activity in area V4. And the interesting thing is that the activity in V4, as a function of the current level, resembles the perception. So if we give about 12.4 microamps, then the activity in V4 is basically half maximum. So we can basically use the activity in V4 to see whether the stimulus is on its way to reach conscious perception. That's very useful. Now, another important question is, if you then stimulate one electrode, the animal is seeing a spot of light. What happens if we stimulate a pattern of electrodes at the same time? So to find out if the monkey then perceives a pattern, we trained the animals to report 16 letters of the alphabet. Okay, so we present the letter D. Now, you can't ask the monkey, what did you see? So we had to train the animal, and we trained him to make an eye movement into this choice display. Okay, so if the animal saw letter D, we expect him to make an eye movement to the letter D in the choice menu. And now the critical experiment is we're not going to present this visual stimulus anymore, but we're going to stimulate a pattern of electrodes, okay, aiming to recreate a letter by directly interfacing with the brain, hoping that the animal will report the corresponding letter. Okay, and this is the result of this experiment. All these green dots are locations where we can create a single spot of light, of phosphine. But now we're going to stimulate multiple electrodes at the same time, trying to create a letter. And the blue dot is where the animal directs his eyes. We try to create.
You have a minute?
Yeah, I'm almost done. So here,
Okay.
the electrodes, the electrodes are stimulated in the shape of a letter A. Now it's an L. It's a T. Okay, so this is very exciting to us. It's the first time this has been demonstrated that pattern stimulation of the visual cortex can give rise to pattern perception. Okay, so this is an important step towards this dream of creating an interface with the visual cortex of blind people. And we hope to be able to go into humans in 2023, although don't be too angry at me if we can't make this work. Okay, so what we demonstrated here is that variations in consciousness of weak stimuli are related to failures of propagation from lower areas to higher areas. And this knowledge will help us in the design of a visual cortical prosthesis.
Thank you. Thank you. And I was only giving you time signals because we have so much activity in terms of questions. I have one, and then I'm going to invite your colleagues back. I mean, so clear the end result, the importance of your work. That's critical when we talk about practical applications and medical use cases. But there's a science fiction element, isn't there? Of course. I mean, if we're talking about putting AI between a camera and a brain, and I talked ethics with Mavi at the outset, what do we need to be careful about? Should we be wary?
Yeah, so there's definitely an ethical dimension here. So there have been patients that have been implanted with electrodes in the visual cortex. And the simplest thing you can do is simply look for contrasts and present the contrasts in the visual field, actually on the brain, where they are being detected. That's a very simple algorithm. But you can also make it more sophisticated. Suppose you're a user of the prosthesis and you want to cross the street. You don't want to see the trees, you want to see the vehicles, right? So then you might only want to select those electrodes that correspond to vehicles. It can be useful, but of course there's an ethical dimension here because now the algorithm is deciding what the subject will perceive and what not, right? So therefore it's also useful to have these ethicists on board within the Human Brain Project.
Thank you very much. And as I see, people are rife with activity and interest. So let me please ask Mavi and Marcello to join us. That would be fantastic. Now, I am going to, in a second, open up directly to questions from the floor, but I just want to give Mavi a chance to in a nutshell, come back. You said, within the project, you focus on these two aspects of consciousness. They're measurable. We've just heard about the concrete examples. Anything else that you wish to comment on in terms of those medical use cases or the practical applications? Anything important for our audience to understand?
Well, I think it's very important. You were asking me before, consciousness sounds slippery. How do you grab this? And we have seen two very concrete medical applications of consciousness studies, but something that I find very relevant is that they are very closely linked and very connected to the basic research, both of them. And I think this is very important because it's an excellent example of how basic research translates into medical applications. And also the fact that by the application itself, we learn more about how consciousness is generated and how it functions. So the application gives us also basic information. I think you are muted.
Sorry, I just, as you said that, I saw it. I'm so conscious to not let my paper shuffling interrupt all of you speakers. So I'm going to pass directly to questions that have been coming in. I think we've got some from Marcello first. He was our first speaker. So here we see from Andrew. Can you see that one, Marcello?
Yeah. I see it and it looks interesting. Quick answer. It's a very good question, and epilepsy, it's a very good example. So epilepsy, it's a condition where you have global activity, global gamma activity, a strong ignition of gamma activity, which is hypersynchronous. And, we thought that gamma activity and gamma synchrony was super relevant for consciousness, but actually during seizures, you have this explosion of gamma activity and no consciousness. Why? Probably because you have a loss of differentiation and low complexity. So it would be very interesting to test what happens in terms of perturbational complexity in epilepsy, but we know already that, activity is less differentiated and more stereotypical for sure. So that fits with complexity.
Okay, thank you. And of course, any of you, I know this is directed specifically at Marcello, but if either Mavi or Peter have something to add or to slightly take the question in another direction, you are absolutely welcome. I'd like to know if the PCI is binary or if it reflects consciousness in a graded manner.
Yeah, great question. Thank you, Benedetta Cecconi. Thank you very much. So it is gradual. Of course, it's a continuous, but in medicine, you need to apply thresholds and cutoffs for practical purposes. So this cutoff that we set is not the magic boundary between consciousness and unconsciousness. It's just a medical threshold. Above that, even though you don't behave, you don't respond, I have to suspect that you are conscious. But it needs to be taken with caution. And, the calibration is not perfect. There are many limitations, but if you are clearly above, it means that we see more than what's evident in behavior.
I think we did have a question for Peter, but I don't want to cause problems. There we have it. There we go, Peter.
Yes, that's also a really cool question. Actually, in the paper, we looked at that and we investigated whether, if you give a very brief stimulus, whether the brain state before you present the stimulus predicts whether the animal is going to perceive it or not. And indeed, we found that if you look at the cortical state, so say the degree of the different types of oscillations, predicts pretty well whether the brain is kind of ready to perceive something or in a state in which maybe the brain is more noisy and a weak stimulus is more likely to get lost. So the answer is yes, that plays a role. And if it's specifically about the face, in our study we did not look at that, but other studies have looked at that, like Pascal Fries. And I think generally they find that the face also plays a role.
Thank you. I know that there's more questions coming in from you, Peter, now, Peep. So we'll stay with you just for a moment. How long can the electrodes remain implanted before they are rejected? Could that be a challenge?
Yeah, it's a really important question. So right now we are using these so-called Utah probes. They are kind of arrays with needles. So they're basically silicon needles. The ones we are using now, you have 64 of them arranged in an 8x8 array. You punch those into the brain, but they get rejected typically after a year, sometimes 2 years. So for a blind person who is implanted, that would not be ideal, right? So you just get used to your system, and after a year or two, you start to lose your sight again. So that's not a way to go. So we're actually working with many partners on other types of electrodes. One candidate that is highly promising are very thin and flexible electrodes, for instance, made of polyimide. And the first impression is that if you implant those in the brain, they will stay good for a long time. Of course, the jury is out whether they will last for 10 years or 20 years. So that's an important scientific question to be addressed.
Peter, can I just stay with you? And I'm just hijacking for one quick question, because we were talking at the beginning about how your work is about writing to the brain. So you're creating perception. So I just want to tie off that presentation. If you stimulate higher visual areas, can you add functionality? Can you create richer perceptions?
That's also a very exciting question. So if we stimulate in V1 and we don't have a lot of control over the precise cells we stimulate, electrical stimulation is a coarse thing. So you stimulate a random subset around your electrodes, maybe 300 micrometers, so less than a millimeter, but still a wide variety of cells. So therefore, you'd have no control over the color of the perception. So one exciting idea could be to also have electrodes in higher areas, say areas where neurons code for colors, so that you could also color in the picture or give depth or all kinds of other properties that are not so easy to control within the primary visual cortex, but what would be the first and most logical place to implant.
Thank you very much. I'm just giving the team, because I know they're dealing with a lot of questions coming in, to be able to post another question. So if there is another question for either Peter or Mavi, or here we go, or for Marcello, here is our next question. Marcello.
Yeah, I think this is for me. Yes, you can. Applying fractality analysis, it's been done. There's a paper by a Spanish group collaborating with us, and I'll retrieve the paper and I could send it if I have a contact. So yes, any measure of complexity works in the end.
I think we have a question that anyone here could come in and answer. So you can see that, I think, ladies and gentlemen. Should computational neurobiology somehow guide research on this high scale level? What would be useful from this branch? Who would like to?
Well, I can start. Oh, sorry.
Yeah. Go ahead, Mavi.
Mavi, come in.
Yeah. Well, yes. Yeah, computer models are very critical for this, because, as I mentioned before in the introduction, they allow us to bridge all these different levels. This is our bridge to go from dendrites to the whole brain, is to use models, and also models allow us to explore much larger parameter spaces that we can do experimentally. So therefore, with the models, one can make predictions that then they can be tested experimentally. So therefore, it's very useful, even at this very high level, to use models, make predictions, and evaluate. Yeah.
Peter, do you wanted to come in there?
No, I think what Mavi said is very true. So there's important work to be done for modeling. For instance, I said that suppose you want to cross the street and only want to visualize or present the vehicles. That requires sophisticated models, because you have to know which pixels in the street belong to vehicles and which pixels not. So also there, these computational models are very important.
Anything to add, Marcello?
Well, it's what we are going to focus in this phase of HPP largely. So it's really, we have all this data, we have curated the data, and we are ready to integrate them into models. And I think models are going to be crucial, really.
I think we have enough questions to keep you guys on screen for up until midnight. So I think if our team behind have got another question that are coming in, I can see, there we go. There's a lot of activity. Marcello, for you.
Okay, so this is related to the other question about fractality. So once you, perturbational complexity index is based on algorithmic complexity. But once you have this pattern of interactions in the brain, either you can apply fractality, you can apply criticality measure, and they all sort of work. It's a very general concept. And there are beautiful papers. I will, again, by an American group, I think, about deviation from criticality in anesthesia. And this is a great paper. And I think, if I remember, well, it's, well, I'll send the paper again. Sorry, I don't have it, but it's there. Criticality matters. Yes.
Thank you.
Complexity.
Can I just say, do you know what's so fabulous, Marcello, with your Italian? I love the way, genuinely, you describe others' work as beautiful, beautiful papers. It's so stereotypically, as I would imagine Italian would speak about his own and other people's work.
Yeah.
So it's beautiful work.
Yeah, I can do that.
It's lovely. It's lovely.
Yeah.
It's so important because we're talking very technical stuff here.
It's true. I mean,
Peter, here, good question. Could a chemical instead of an electrical interface be considered?
So I think it's really good to open the scope here. So electrical stimulation has its limitations. You have little control over precisely the neurons you stimulate. Chemical stimulation, it depends on what you mean by it. So if it would be like adding chemicals at specific locations in the brain, that could be a bit too slow of a process. But one way that people are now considering to stimulate is optogenetics. So you kind of add channels into the membrane of a cell. So in a way, that's also a sort of chemical stimulation. And you can open them up with light. The advantage of that method is that you can really stimulate with a precision at the level of single neurons. And so last year, for instance, by the group of Carl Dijsselrood, they demonstrated that they were able to stimulate multiple single neurons, and they could precisely control which cells they stimulated and which ones they did not. Now, it has many advantages because this precision is fantastic. It has a few disadvantages as well. So the methods to get this light stimulation at the sufficient precision require a 2-photon microscope, which is not very practical to carry with you when you're, say, walking around in a city. So there's some room for improvement there. And also, the way to a clinical application may be a bit longer because you would first have to treat the brain of a patient with viruses to implant these channels into the neurons. So it's definitely being considered. There are clinical trials ongoing, but before we have a working product, a working treatment that is available for many patients, will take many more years.
Okay, let me just give us all because I would like to bring Mavi in briefly here, please, as well. It's we have 7 minutes, ladies and gentlemen. I'm just conscious of timing. If we can't answer every question, I am sure we can filter questions your way with your permission. I just want to ask, Mavi, you can see there's so much interest, so much activity. Are you surprised by these questions? Are you warm, encouraged by these questions? What do you sense from people's response to these presentations?
Well, this is something I encounter always with, with the general public and also, of course, with the specialized. But there is a very large interest in brain research, and this is the truth for general public. And I think we are seeing this. I would like to add up something, a small thing to the last question about the chemical stimulation of the brain, to bring forward the work of someone in our group, in our work package, Pau Gorostiza. He works on developing this photopharmacology, which are drugs that are sensitive to light, and they allow also a control, spatiotemporal control of activation of neurotransmitters. And I think eventually they could be also useful for the kind of visual prosthesis that we have seen.
Thank you. I think we have still time for 3 or 4 more questions. If you, ladies and gentlemen, can be as concise as possible. Here we go. Here's the next one.
All right. So let me say, I found that beautiful paper It's loss of consciousness associated with stabilization of cortical activity. Last author, Alex Proecht. Very nice paper about criticality. Now, this question, so if I understand it, we have, as I mentioned briefly in the talk, we think we have a couple of mechanisms that we think they're responsible for loss of complexity and interaction among neurons. One of these mechanisms is called bistability. It was basically discovered, Mavi contributed heavily to the description and discovery of this mechanism many years ago. I remember reading Mavi's paper in 2000 and being enthusiastic about it. And now it's very important because this means that neurons, they receive an input under some conditions. In sleep, this happens, but also after brain injury, they activate and then they become silent. And when they do that, everything is lost. Interaction, feedbacks are lost. So we think that this is important. And we think that in many conditions, circuits after an injury, they enter this bistable dynamic, which is a functional state, potentially reversible, and you can optimize network complexity and restore network complexity by killing and preventing bistability. But there are also other mechanisms like dendritic coupling that we need to explore, the one that I mentioned. So yes, the goal is to restore and optimize networks.
Perhaps we have time for one last question for each. So here is yours, Peter.
So the question is whether it's electrical stimulation or fMRI. So if you want to stimulate the brain, fMRI is not a method because it's a method for reading brain activity. So there is, of course, a magnetic way of stimulating brain activity as TMS. So Marcello also already talked about it. And if you do TMS kind of to the back of the brain, subjects will see many subjects will see light perception, but it's kind of a very large blob of light, a bit diffuse. And the reason is that you're probably stimulating many neurons across a large region of the cortex weakly, and it doesn't have the spatial precision that you need to really get the very precise perceptions. So the next level is to put electrodes on the top of the brain. Then you get somewhat smaller perceptions, but only if you put a very tiny electrode into the brain, then you get these very, very precise perceptions from which you can build letters.
Thank you. And a last question here for Marcello.
All right. Yes, I'm aware that in these states, psychedelic state, there is an increase in entropy. So entropy is a measure of complexity, but I have to stress that one thing is to measure complexity on spontaneous activity. Another thing is to measure it on after perturbations, because perturbation makes sure that the complexity you measure comes from interactions, causal interaction. If you measure it based on spontaneous activity, sometimes you can have high complexity, even for random generators and independent sources. So we need to be careful to equate these two ways of measuring. But indeed, the finding of high complexity under psychedelics is very interesting, high entropy, say. But we need to be aware of the differences between measures. Yeah.
Thank you very much. I'm going to close questions there. And again, I am sure if you have a burning question that you need to pose, you nod, gentlemen. I don't think you would want to be inundated, also Mavi, but if people want to get in touch, I'm sure they can go through the Human Brain Project to reach you, because that is the purpose, and to widen the community. So thank you so much. Mavi, may I start with you, please, and ask if there is any last takeaway for our audience? You are very proud about the project. There are some unique aspects of the Human Brain Project. Is there anything that you didn't get a chance to say? Now is the moment.
Well, I would like to say that we live now through this time where we mostly think about a virus that is very challenging, but really from the perspective of brain research, I think these are very exciting times, because as we have here, we have tools that we never had before. We can study the brain, but also we really need to study the brain. I think this is very important. The prevalence of neuropsychiatric diseases are very high. They are not getting less because of the virus, so they still are there. We still have to keep working on this. And also, of course, we have to understand ourselves. We have to understand nature and ourselves. So the key is in the brain. And therefore, I think, well, I really hope that the Human Brain Project and and the research done in it and e-brains is going to help to make a difference for the whole community.
Thank you very much, and thank you also for bringing in the context there. And of course, it doesn't need me to tell you three that it's science, science, science. If I hear one more time, follow the science, I mean, that is what everybody is saying. It's a curious time as well. So tell me, I mean, you two gentlemen, what does e-brains mean for you in your science? The last word from each of you. Marcello, please.
Yeah, I mean, for me, it's been a unique opportunity and a surprise, because when I entered HBP, I had a project, I had in mind some ideas, but I didn't know what was coming and how this was building up. And it's becoming better and better. And I think now really we are in a very exciting phase. So we didn't have time to go through all these things, but the idea of putting together all this data in a community and then using this platform to do something that can be useful to other scientists, and then data sharing. So I think that at some point we'll have so much data, well organized, that many questions can be answered even by people from outside HPP. And this is great. I think it's great. I can only say beautiful.
Bellissimo, bellissimo. Yeah. Peter, I think you were nodding at some of that there.
Yeah, I would like to join Marcello in saying that this is a great facility. So we are heavily using the possibilities to publish data, to share data with the world. And also for us, for this project, the brain atlases that Human Brain Project is making are really, really important. If you want to interface with the cortex, for instance, to stimulate or to record, you really need to have a very detailed map of where are the cells you would like to stimulate. So there, e-brains is also tremendously helpful.
Thank you very much. It was very, very important to bring in about the brain atlases at the end. So thank you for flagging that up. Thank you very, very much to the three of you for coming and distilling the contents of your considerable brains with me and with the audience, and for responding so rapidly to all of those questions that came in. Do stay on the screen, because I'm going to wrap in a very, very short time. I'm going to thank, of course, the technical, the comms, the digital teams for all of their hard work in organising, hosting this webinar, and of course, as we say, keeping the lights on and us all online. And the biggest thanks, of course, goes to you, ladies and gentlemen, for turning up and for tuning in and for being so prolific with your questions. That's fabulous. If you loved this, don't forget to join us again in 1 month. We are going to be looking at brain-inspired computing. There'll be 3 other heavy hitters with whom you can interact. And of course, before then, you can also share all your thoughts, your enthusiasm, and your words of wisdom, #BrainMatters. And on that note, I think my fellow speakers here can only thank you for being with us, to wish you a very, very good evening. Take care, be safe, hopefully feel you, because we won't see you the next time. Thank you. Good evening.